Okay, we'll start with some questions.
It's Greg Burke from Wake Forest. I have a question for Rory with regards to what I'll call ancillary studies. I'm over here. Technically on. So once you develop the infrastructure, 500,000 folks, there's interest in extending from that original phenotype into other phenotypes, and you've done that with subsamples. I'm curious sort of your strategy for both making decisions about lots of good ideas that come your way, and prioritizing those in such a way that you move forward with it. And the second was your decision to keep everything central, and whether how that has benefited you as you move forward.
So with respect to how we enhanced it, we had an enhancement working group that had information being fed into it from various experts in different areas. We would explore the proposals that came to us, and wherever we reasonably felt we could introduce things, we tried to do so. So we tried to be facilitatory because we were aiming to build a resource that those researchers wanted to use. Obviously, it depended on getting the funding, so we would work with them to put together proposals. But in fact, every single enhancement that came through the enhancement working group that we then went to the funders with, they did support. There were areas where we held back. We had some meetings some years ago with people from NIH around environmental monitoring, and we weren't clear quite how to do it at scale at that point. But in fact, our enhancement working group is now going to look at that and consider whether that's moved on such that we could do it at scale. And the other thing is to look at what else we could do in the imaging visit. So we didn't know we were going to have an imaging visit. When we proposed it to the imaging community, they said, great idea, but you didn't mean 100,000, you meant 10,000. And we said, no, we meant 100,000. And now we've got to the point where we might do that. It's important not to forget the opportunity to get more sample or more kinds of measurements. And in fact, it's being used as the basis for the UK government's dementia platform. And so the plan will be that 10,000 of the individuals who are imaged will get a repeat image a few years later. And we're doing much more cognitive assessment in that imaging visit. So we're trying to facilitate. In terms of the centralization, I think this, as I said during my talk, the separation between what should be done, which is an academic pursuit, and how to do it at this scale, which I think is an industrial pursuit, separating that worked very well. It required less of the academics in terms of having to turn a handle for 5 years. And one could really get very substantial efficiencies in how the process was done, improvements in quality by ensuring it was done in a systematic way. The point about sample tracking, all the samples were brought into a central facility where they were processed using robotic systems with limb systems that tracked according to barcoded tubes, so that one can get to the particular tube in the particular person very rapidly using robotic storage systems. So I think you can get big efficiencies that allow you to do more, and you can get better quality data at the same time. So I don't think there are losses.
That certainly speaks to the need for public-private partnerships.
So the system, which, as I understand it, a lot of people are using, of doing the exome sequencing in a research environment, and then when you identify variants that you judge to be important, typing those in a certified environment, getting it into the EHR, that of course works, but it leaves a lot of information, as it were, on the table that clinicians might like to have access to for hypothesis generation, including hypotheses about management. How big a constraint do you think that is right now? And currently, is there a way that clinicians can explore the broader genomic data to generate hypotheses about management that they might have? without going through the path of certified typing? And if there isn't a way to do that, does that itself, in your view, constitute a really strong argument for moving this initial sequencing into a certified environment sooner rather than later?
Yeah, so you've, again, mixed the clinical needs and the research environment. So as long as it's within a research environment and the clinician is in the research environment, then you can explore the data and follow up and confirm and communicate results back to the patient. If the clinician is outside of the research environment at the moment, that would be a handicap of the not full CLIA implementation part of our system. So for me, it would be a big benefit if all of the data was generated. I mean, the other big problem at the moment with doing it in a research setting, we're able to confirm the positives and make sure they're not false positives, but of course we don't have any quality control over the negatives, and we don't know what false negatives there are in the data.
A question for Rory. In terms of, as looking for this cohort, what were the thoughts regarding not including children and really staying in a pretty strict group of adults?
Yeah, there were a lot of discussions around the age range. Should you go younger and get people before disease has developed, but then have to wait longer for the disease, or to go later in order to get quicker wins, but you might be measuring things in people where disease is affecting the things you're measuring. So the 40 to 69 was a compromise. I think that the kinds of samples and questions that you're likely to want and the kinds of diseases that you would be wanting to study in a cohort of children and in a cohort of adults may well be different, and that it might then be better to have separate cohorts that focus on those particular areas. And that's the strategy that's occurring in the UK. So the LIFE study that Biobank is helping with, based on our industrial research expertise, being run by Carol Deserto, is a study which will be about 80 to 100,000 mothers and their children and the fathers.
Okay, thanks, Antonia.
Antonia Calaf at CDC. We haven't talked much about using the cohort for evaluating exposures to environmental exposures by measuring biomarkers. And I was wondering in the UK, in the biobank, then if it has been used for measuring environmental biomarkers.
Not as yet. We have samples that we hope that people will think of to use it for that. We want to look again at the possibility of measuring various exposures that the participants have in terms of exposures in the home, things like radon, but also the other kinds of exposures that were described earlier in the day. But there haven't, I mean, what samples are there. If there are samples that are on the list I put up that are not there, then this is a really good time to tell us, because if we're going to image 100,000 people over the next 5 years, we could get additional sample. And so, for example, we haven't got very much in terms of samples that would be able to look at immune response, and I think that that's one area that we might look at. Should we be getting feces to look at the microbiome, et cetera? We would really welcome input now, because our mantra in Biobank was defer. You do what you had to do now, and defer anything you didn't have to do now. And then once you've done the things you had to do, then think about the things that you'd like to do. So we didn't worry too much about the access procedures until we actually had something to access. We didn't really worry about the follow-up until we had people to follow up. And I mean, I know it sounds obvious, but if you try to do everything, you're likely to fail. You really need to focus on what you have to do, and then think about enhancements. And so, we have opportunities. We would welcome suggestions.
Try and call that an agile approach, I think. Jeff.
Thanks. Jeff Ginsberg from Duke. Rory, you may have mentioned this, so forgive me if I missed it, but to what extent is industry a supporter and have access to the biobank? And If that's not the case, why not? And if it is the case, how's it going?
Yeah, there have been very few requests to use the UK Biobank by industry so far, although we have explicit consent from participants for academic and commercial uses, and we've done consultations when we finally got round to writing the access procedures with participants about their views on that, and they were very positive, particularly when one explained the rationale for where do you think drugs come from. And so we haven't as yet got industry involved, and I think it's something we're working on now that the resource is mature. So in this next quarter, we'll be releasing the genotyping data for 150,000 people. I think that will stimulate more interest in using it. During the next year, the biomarker data. We've got the linkage in place. So I think it's more useful now, and this is the time we hope that industry will start to use it. I think there's also more than one industry. So it's not just the pharmaceutical industry, I think it's a bioinformatics industry. How do we turn complex data into usable information? How do we get automated analyses of 100,000 cardiac images? Not measuring the things we know about, but doing reverse, the analogy to reverse genomics of comparing the disease against the images and working out what in the image is related to disease. That is a massive informatic challenge that we need the industry to involve in.
Thanks. I think we had Elliot, is it? Mike, and then, sorry.
All right. My name's Elliot Margulies. I'm at Illumina. I enjoyed both of your presentations a lot. And I wanted to address particularly, David, you have a concept of an individual wearing a research hat or wearing a clinical hat, and the distinction between the two. But one of the things I'm trying to figure out, and I think we all are, is how do we combine one ecosystem that can serve both a research community and a medical community? And I wanted to, I'll start it with a specific question around something as simple as, a patient identifier is appropriate for a physician to see, but not for a researcher to see, or the same physician with their research hat on. And have you built systems that are handling things like that right now?
So this difference in culture and style and other things remains an impressive difference to me. And when I'm talking particularly to companies who are coming to sell me equipment or products in my lab, I always have to stop them and say, you're usually selling to researchers, aren't you? Because you need a different person talking to a clinical lab person. And I make the analogy, it's like having a Mac or a DOS brain, an operating system, and very few people have both platforms and can fastly go back and forth between the two. So it's usually clinical and research marketing people, clinical and research language. From my biased percentage, people who understand the clinical part, it's easier to learn and understand the research part and the differences, but that's just my personal perspective and bias. I think it's projects like these where you have to make sure you have representation and you at least acknowledge the differences and see if you can negotiate some middle ground or agree that when the two are different, you always go to the higher standard or something like that. So it's plays out and is resolved to different degrees of success at different institutions. But the remarkable thing is how separate those two communities continue to be at the great majority of university academic medical centers, and even in a healthcare system, the separation of our research people and our clinical people.
Someone's into Unix. This is not a good analogy. Okay.
Mike Lauer from NHLB. I enjoyed both of your presentations. This gets back at Saker's question from yesterday. You've both built up these fabulous resources. Could you tell us whether at this point you've had some major victories, some major discoveries that have been made and published that could only have happened by virtue of the fact that you built these?
Actually, I can say the reverse. I think UK Biobank hasn't done anything. A prospective cohort is a 10-year investment before you have any possibility of anything happening. And When I was asked to take over UK Biobank, I didn't take up the head of communication post that they were about to appoint, because I wanted us to have a low profile for the next 5 years at least, until we had done something, which was recruit 500,000 people, and then to slowly build up. But I think it's only now that it's a resource that is of any value for research. I hope that it will then generate something, but certainly not yet.
Speaker?
Yeah, we're also very early in just getting the exome sequence data back and looking at it with Regeneron. I mean, there's tons of interesting preliminary data that we think will lead to great discovery research and inform both of our organizations. I think for me, the success will be when there's something that we think we will be able to implement in a clinical real-world healthcare setting to the benefit of our patients. George wants to comment.
Yeah, I agree with David that we're really looking at this as a long-term process, and on both sides, we either want to deliver better health care with better results to patients, or therapeutics or diagnosis and so forth. But just to answer that question in the very short term, when you have somewhere on the order of 50,000 exomes linked to health records, it's enormously helpful in everything else that you do. And I've talked with a bunch of you in the audience who are already trying to do these sorts of things. But for example, we've already identified many, many more variants that we see. Let's say if we're doing a family study and we have many candidate genes that involve novel variants that haven't really been seen before in the small populations that are available. We've already been able to eliminate many, many of the candidate genes and focus only on the right one, because we know in the Geisinger population, we see them in normal people who aren't suffering, let's say, from those diseases. That's one example. Another immediate hit, as Dave said, what you have to recognize is they've already have the cohort information. They've been doing this for 20 years. So working together, we actually define very interesting populations that we're going to now focus on genetically, which you already have, which you already get. So for example, they have a great resource in the obesity world. So they have people who have been obese for years, who they've characterized every metric, their glucose, their lipids, everything else in addition to their body weight, and then they get bariatric surgery and they lose 50 or 100 pounds. Some of them start out totally normal in terms of, let's say, diabetic parameters. That's an interesting population. They have huge BMIs over 40 or 50, yet they have no diabetes. Other ones, they get the bariatric surgery, and there's no change in their diabetes. And other ones dramatically improve. So they're already able to identify which subsets of patients we want to, might focus in for specific experiments. So both in terms of eliminating noise in all of our other work, or defining very exciting projects that could really lead to, important findings, it's already here, it's already now, it's already happening.
All right, thanks. Sika?
So, Sekath Rayson, Mass General. So I just wanted to really say to Rory that the work is truly inspiring. I think it really speaks to the feasibility of doing something like this and the potential value in terms of a bioresource for the nation for the next 50 years. And I want to describe also my own experience in trying to access UK Biobank. I think I'm one of the, 149 investigators who have been approved to the main application, and it was remarkably easy. We put in an application. It took a few months to go through each of those processes. We're fully approved, and I think in about 2 months, we're going to get phenotype and genotype data for 150,000 people. And that ease of access was really spectacular. So I think it's a model for what we try to accomplish here.
Two have obviously been talking together. Terry?
Rory, I wanted to ask you your comment about not having many people accessing the resource outside of sort of academics in the UK, and contrasting that with the multiple requests that you get for collaboration where you say we don't collaborate, it's your resource. So sort of more as a lesson for us, if we build a resource like this, we'll probably have even more of a problem because I think there's increased funding pressure on investigators in the US to have funding in order to be able to do analyses and that sort of thing. So could there be a model where it's sort of collaboration light, where you kind of, pretend that you're collaborating, but really kind of lead them down the path of, okay, you open up this website and you help, support them a little bit, but not because you don't have the staff, obviously, to do a lot.
No. By saying we don't collaborate, I'm saying that we certainly help people through the process and help them to understand the resource. We respond to people's use of the resource and the questions by trying to put more and more information up there. And we do have a team that helps people not just police the access system, but also help people through it. And despite what SEC says, I think that we can make it a lot easier and a lot more streamlined so that people can, initially, all the data that were provided were tailored to the particular researcher, so they had to pick out particular data. We're trying to be much more, here's a set of data. So I think we can provide a lot of support. By saying we don't collaborate, I'm saying that UK Biobank is not part of the piece of research. Other than to say you've used it, that's all that's asked of people. It's their resource. It's their resource to use. Our job is to help them to use it and help them to make it better. One suggestion that has come is that we have UK Biobank research fellows, and that might be a way of training people, based not in UK Biobank, but based in research groups. They could apply to be fellows, and they would really get to know how to use the resource, and that might be a good way of spreading the word. We haven't yet implemented that, but it's a suggestion that came actually from Andrew Hattersley, who's on the board of UK Biobank, who's a genetic researcher.
And maybe if I could just quickly
Sure,
follow
go
up.
ahead.
So maybe something that we could think about is, could we list a list of questions? I mean, obviously, when you get diving into these data, you see things that are really interesting. It's like, gosh, it would be really neat if we had a graduate student who could pursue such and such. And I wonder, is that being too directive, or maybe it might be useful to sort of say, not suggesting it for Biobank, but more for us. If we say, here are some things that really need to be addressed, and wouldn't it be great if somebody would do it?
Yeah, and we're very keen that it's used by graduate students to support master's or DPhil program, PhD programs. The fees are minimal. We're required to do them in order to avoid what we call value-added tax. But we're really, we cover the minimum of cost.
Rex, I think that will have to be the last question, except for the one I want to ask.
So, David, I'm interested in the CLIA problem. So if we think about this cohort and the fact that I think we want to really be able to think, as I said earlier, about implementation as we go forward, what's the added additional cost to do some of this in a CLIA environment? And is the cost such that we'd be better off not worrying about CLIA up front and reconsenting and resampling people when we identify people that we want to actually do something with?
I think the difference in lab practice and quality performance in a large-scale sequencing facility and a large diagnostic lab is much less than the difference in practice and quality control in a small research lab and a small clinical lab. So I think it would be relatively modest costs for existing large-scale sequencing labs to become CLIA certified, and that with partnership with existing expert CLIA laboratories at the same institution or by consulting, could make that conversion to a CLIA certified lab. And there's no problem with sending samples from one CLIA lab where samples are obtained to outsource to another CLIA lab to generate data. So,
but I was interested in the original collection. So in the original collection of 1 million people, would we be better off doing that in non-CLIA and then just reconsenting people where we wanted to do some kind of return of results, or
for me, that seems highly inefficient as opposed to starting the process in a CLIA environment. And again, our blood collection system and most large healthcare systems, blood collection is very efficient, and the added cost of getting an extra tube for the biobank purpose is pretty trivial. So.
Okay, thanks. I'll ask you my question offline. So let's thank both our speakers. Thank you.